In the previous two videos, we touched upon oligodendrocytes and multiple sclerosis. We reviewed how in the central nervous system, most of the axons are wrapped with myelin and myelination occurs from oligodendrocytes, which is essential for efficient transmission of impulses along the axons. For this video, we will be discussing specifically the role of oligodendrocytes in multiple sclerosis. To briefly review oligodendrocytes from the first video, during the development of the central nervous system, oligodendrocytes are derived from oligodendrocyte progenitor cells. However, these OPCs still persist in the adult central nervous system. These adult OPCs contribute to myelin remodeling in physiological conditions and also contribute to remyelination after demyelination injury. In this figure, you can see that oligodendrocytes are derived from OPCs, which are derived from neural stem cells. This process is driven by multiple factors and pathways, including the oligodendrocyte transcription factor 2, homeobox protein NKX2.2, SOX10, and myelin regulatory factor. These are noted with unshaded bullet points as you can see in the figure. The different stages of oligodendrocyte development also have different development-dependent lineage markers. These lineage markers are shown in the orange box. In MS, inflammatory demyelination is driven by the autoimmune attack, which is characterized by the recruitment and reactive activity of astrocytes and microglia. This leads to demyelination and axonal damage. The astroglial proliferation and depletion of oligodendrocytes then result in a glial scar, which is shown in the next figure. In MS lesions, the differentiation process from OPCs to mature oligodendrocytes is impaired. This is due to the inflammatory microenvironment and the presence of inhibitory molecules and causes inefficient remyelination. In experimental inflammatory demyelination, OPC differentiation is inhibited by effector T cells and interferon gamma. The failure of axons to remyelinate is partly due to the failure of OPCs to mature and remyelinate axons, and also due to the death of injured oligodendrocytes. Despite the various challenges, researchers have been looking at strategies to modulate OPC differentiation to enhance remyelination. Although we will be discussing the possible therapeutic strategies in a later video, some approaches include the immunoglobulin-like domain-containing protein 1 and oxidative phosphorylation. Both approaches are still being explored as remyelination-promoting therapies that target the inflammatory oligodendrocyte and OPCs in the early remyelination phase. To force successful regeneration, the potential pathways include galectin-3, estrogen receptor ligand, and the NMDA receptor. Recombinant galectin-3 can promote differentiation of OPCs to oligodendrocytes by activating AKT kinase and suppressing the extracellular signal-regulated kinase 1 and 2. Additionally, the differentiation of oligodendrocytes requires reorganization of lipid metabolism, including the biosynthesis of cholesterol, which is a major component of myelin. Researchers found upregulation of genes involved in cholesterol synthesis in oligodendrocytes during the remyelination phase. When estrogen receptor ligand increased cholesterol synthesis in treatment, a potential target to enhance remyelination was indicated. Lastly, in the EAE model, researchers found that early axonal damage may precede demyelination, which suggests that loss of metabolic support from oligodendrocytes may cause demyelination. Oligodendrocytes and OPCs use aerobic glycolysis for the biosynthesis of myelin and to maintain cell function. Therefore, activation of the NMDA receptor can support axonal energy metabolism by increasing the utilization of glucose in oligodendrocytes. This is because during stress, oligodendrocytes use ATP for cell survival rather than for myelin production.